One of them larger,
Hold there.
and one that goes to a smaller size. As far as we know, there's only one in Guam, but I'll have to check. The people doing the revisions on this initially thought that the one in Hawaii and the Central Pacific was different from Edilus carbunculus, and took the next available name for it, Edilus marshii, and that's been used in the recent past couple of years. However, there's a brand new paper that came out just a couple of months ago that established that Edilus, the type of Edilus carbunculus, the specimen that bears that name, from way over in the western Indian Ocean, actually is the same as the species in Hawaii and probably Guam, that's Edilus carbunculus. So Edilus marshii, which had been used as the name for this fish, isn't used now, and that's a fairly recent thing. I don't know if Chris has seen that paper, but I can pass it on. And the larger species that occurs in the Indian Ocean is down through Samoa, but I don't think it occurs up here. Doesn't have a name yet.
Great. Thanks, Bruce.
Yeah, I'm embarrassed to admit that I have the paper, but I haven't read it yet.
Watch these, Nav.
Go ahead, Nav.
Yeah, so the names have changed back again to protect the guilty.
Go ahead, Nav.
1 hour remaining on the bottom.
Great, thank you. So Alex Kerr from Underwater Guam and Guam Marine Lab has said that the Edilus in Chamorro, known as Abuninas. Abuninas. I hope I pronounced that correctly. I'm sorry if I didn't. Thanks for having faith in me, Dale. And so, Abuninas actually means, Alex is continuing with the brilliant bits of information, Abuninas actually means the stomached one, because when it's brought up to the surface, its stomach comes out of its mouth. And that is, of course, because fish have swim bladders, which are filled with air, and the change in pressure when they're brought to the surface, because, I mean, right now we're at 340 meters depth, which is, I think, about 30 atmospheres. Yeah. When they're brought to the surface, they, of course, air expands, and the swim bladders expand to a point where it forces a lot of their guts and insides out of their mouths, and sometimes they even explode. And their eyes bug out.
There's a big shadow there with the craft, huh?
I have nothing to say to this.
It's true. Physics.
Like, really nothing to say now.
Yeah. Especially with the lights in.
Yeah. I see something run over there. One of those redfish.
And I think that's the first time we've seen any of the deepwater snappers on all three of the fisheries dives we've done. So that was a great spot. Well done, Chris.
I'm getting currents kind of turning into port too, so yeah. Quite a bit actually.
I think the tether's going way out
Yeah.
to our right and then kind of rotating you.
Yeah. I think we're ready for a move, Pi.
Yeah, I think so.
Bridge nav. 40 meters, 180 degrees, 0.2. Thank you. Yeah, it's about the end of your leash. turning pretty far too. I'm at 62, my set point's at 80. So.
Yeah. Let me just come way to your left here, if I can make it.
So Eric Cruz, who works for National Marine Fisheries Service in Guam, is here at the IRC Command Center this week, and he told me that they have both of these Edalus species over in Guam, both Edalus carbunculus and the undescribed species. So I'll have to do a quick check on the characters, the field characters, to see which one that might have been.
I was giving you some tugs.
Yeah.
Thanks, Bruce, for all of your fish knowledge, which you continue to share with us and teach us.
Laterals are getting work out today.
85 to stay put.
So, shore-based science and Chris, we have a little bit less than an hour left on the seafloor. Is there a particular plan that you'd like to follow for the next hour? Or do you want to just continue doing what we're doing, which is moving up slope and imaging as things appear?
Well, my vote would be to continue going up slope and see if we could get to 300 meters at least, and maybe we'll get into a resurgent coral community.
Okay. I'll pass that on to our pilot and navigator. Nav, this is Watch Lead.
Yes.
So I was just wondering, I mean, do you think we have the ability to get up to about 300 meters before the end of this dive?
Yep. Yes.
So I was just wondering, I mean, do you think we have the ability to get up to about 300 meters before the end of this dive?
Yep. We should be able to. We're 160 meters from waypoint 2, but we're only,
Cool.
Yeah, we'll be able to get up to 300 meters water depth, I'm sure. But just to let you know, if this current gets much stronger, we won't be able to continue the dive. We're right at the limits of what we can do.
Just curious, how strong is it? Sorry, I just spoke over Deb.
Always.
Just curious, how strong is it?
We can
just a guess.
Figure that out on fins.
Sure, we can try if you think you can recover for more than a second of drifting.
Yeah, I could try to push out. We can do it.
I'm coming forward.
Copy.
So we started seeing ripple marks a few minutes ago.
Okay.
And we still see yet another, just a very small, like, offset
auto heading in
the...
There's some fish over there.
The truest test is nothing,
2 fish to the left, please.
2 fish to the left.
Redfish and bluefish?
I can't quite see over this.
Okay, quick sip.
Yeah, the Edalus like to hang out under ledges too, this particular species, and that is one right there, another one.
Would you like me to come wide again?
No, that's fine right there. What'd you say, Copilot?
Sonar.
Oh.
Couldn't see over a ridge for a second.
Okay, keep coming, video.
Okay, zooming in. There's also a red one that's just going out of frame beneath us.
Yeah, I saw that guy. And yeah, just hold there.
Okay, that's a partial.
Copy. Coming up for you.
So because these are so important for us, some of us have been working here at the IRC. Alan Andrews got a screen grab of that previous Edalus. And for the field characters that we have, Eric Cruz and I decided that The one that we filmed earlier is the smaller species, that is Edilus carbunculus. Eric says that the larger one also occurs in Guam. That was undescribed, but the one that we saw earlier is the small Edilus carbunculus.
Catch him as he comes around. Turn around. Okay. Let's let him go. Come on.
Okay, coming wide. Okay, that's full wide.
Copy.
Current will take you right back here. So. Looks like the ship's almost there or stopped?
Still moving.
Okay.
So what do we think local high is here?
It should be stopping soon.
Close enough to 180 probably. Yeah.
Yeah. That's what bathymetry would agree with too, so.
Is that the... Yeah.
Yep, stopping.
Yeah.
Bridge nav. Let's add another 40 meters due south. Thank you.
So Bob Stern on the event log does bring up a good point. Because the current's so strong here, I think we would expect to see more well-formed ripple marks, but we're not. And so I'm wondering if it's because there's simply just not really that much sediment, and most of that's just cemented down.
Yeah, Deb, when I land, I don't kick up all that much.
Thanks. That's good to know.
So that's not too...
Yeah, so this is really not like a muddy bottom. There's not a whole lot of sediment that's moving around here.
Yeah.
That looks like denser white stuff that way.
Yeah. What is that? Is that another army of brittle stars? I'm going to snap from here video. See if we can bring any of that out.
Okay, zooming in.
Or coral.
Wow. That's impressive.
Okay, so if we could get some zooms to make sure we've seen these before.
Do you want me to go in more, pilot?
No, come wide.
Okay, coming wide.
We're
I
working
thought the
our way
pilot
over
was saying
there.
no.
No.
Yeah.
The ship moves
Yeah,
off
this looks new.
closer over there.
So I mean,
This
we've
looks,
moved through several different zones.
So
Might be able to
Was the last
saddle over
fish
a little
the
bit.
Edilus carbunculus?
But if you want to get us over the middle of it, just do your thing. Stop it.
let me figure out if they
It was
want
an
us to-
Edilus. I don't know if we could tell which species.
It was carbunculus, Bruce. You could see the white tip on the lower caudal lobe.
Watch leads nav.
Go ahead.
We are bucking the end of the tether, so if we actually want to go
Okay,
look at that,
let's
we're
go
going to have
ahead
to
in.
stop the ship and move over there.
Zooming in.
Into the tether.
Yeah, Chris, unfortunately, both of them have the white on the lower lobe of the caudal fin.
Hold on a sec.
The undescribed species has black on the tip of the upper lobe,
keep
and
coming.
carbunculus
Okay,
does not.
I'm going in.
This is as close as we're going to get if you guys want to
Oh,
go.
I thought you were referring to maybe it was an onaga rather than an ahu.
Copy. Just try to do
No,
as
it's
best
just
we can
debating
here,
whether
video.
it's the undescribed species of ahu or the carbunculus, and Eric says they both occur in
the
Guam.
ship's underway going uphill. This is off to the east. So if we need to go over here to study it, this is the best image you're going to get.
Shore-based scientists. So this is the best image we're going to get of this particular set of biology,
Full shot of all
unless
of it.
we move the ship.
focus
Would you
through the
prefer to
current.
continue to move upslope,
That's a
or would
full
you like
wide.
us to stop the ship and come back here?
Just survey the whole scene here.
Well, I would vote, let's get into this bed. This is quite a significant bed, and try to get a better close-up of this animal.
Okay. So Nav, I think we'd like to stop the ship, or move it over here.
I gotta come up a little bit.
Yeah.
Bridge nav.
Okay, so we're gonna follow into this
Can we stop
bed
the ship here?
and see what we find over here.
And then let's go 20 meters, 130, 0.2.
I'm happy.
Copy.
Bridge nav.
18 delta, not a whole lot of tether.
Can we make a move of 20 meters, 130 degrees, 0.2 knots? Thank you.
Okay. Come on in, video.
Okay, coming in.
See if we can pick out one of these. Can you tilt down for me, Copilot?
This is partial.
Copy. Come on down. Let's get the closest one we can. How about the one to the right over there? This guy. Yeah. Is that partial?
Yes, that's partial.
Okay. You want to come on?
Okay,
Keep
coming in. Oops.
Top.
That's max zoom.
Copy.
Down bot. Nice piloting. It's a lot of current.
It's going to make me oscillate.
So coral reef, not coral reef, deep sea coral biologists, what do we think this is?
I think I got a little extra
Very
tether
interesting
there, maybe?
polyp orientation.
Okay. Can we come full wide video?
Okay, full wide.
Let me try to sneak
Coming
in
full
there.
wide.
And yes, there are a lot of
Yeah,
them.
That's full wide.
I think we're still collating.
Copy.
Sir.
Copy.
And not 70.
Copy.
Nothing too bad. Yeah.
Okay, video. Let's come back in.
Okay, coming in.
Tilt down, Copilot.
It's a little difficult to
let's get
gauge.
some at our
What's
feet
the
here.
what's the angle of the
coming
slope
down.
here
Hold
that
there.
we're sitting on?
Holding.
Keep coming down. And hold there. Let's get this guy if we can. Tilt back up a little.
Try. There we go.
Okay, video. Keep coming.
Okay, coming in.
Try to center it for you. It's a little tricky. Sorry, video.
So,
So I'm troubled. I'm almost wondering whether this could be a bamboo coral. I could swear there's some nodes on the dead one, and I thought I saw faint nodes on one of these.
Yeah, thanks. Sorry, I'm asking a lot. What are you looking there?
Max.
Okay.
18 delta.
Okay.
15 altitude.
Is that all you got, video?
yes, that's max zoom.
Copy.
another colony to the left of this that's dead, and only a few live closed-up polyps are on one of the branches, and it might be worth taking a look at that afterward, but it kind of looks like a node to me near the base.
Coming wide.
Okay, we gotta
Let's
go.
go wide.
No, should be stopping. You got time.
I'd like to get out of here.
Okay.
Coming uphill. Sort of translating. May as well, there's a whole field here.
Hey, Nav.
Go ahead.
So are we doing okay in terms of the current?
It's right on the edge.
Okay, thanks.
There you go.
Diva wants to know what the slope is here. Any idea? No.
Okay, we have Scott back. We got his attention by mentioning bamboo, so I wonder if we could do another zoom on one of these things.
We're working on it, actually. Getting out in front a little bit.
Okay. Take your time.
It's hard to.
So,
to
onshore
guess right
scientists,
now.
we're just trying to navigate a tricky situation here. So, as soon as our pilots can, they'll settle down again and get us a good shot.
Okay, video.
Okay.
Let's,
Coming. Oh,
yeah, let's come on in.
okay. Coming in.
Oops, sorry.
You're okay. Coming in more. That's max zoom.
So, onshore
that.
scientists, what are you thinking? Scott's guessing paragorgid or anthothellid. Oh, and there's also a really nice little shell there. I'm not sure if it's a gastropod or a pergurid. I'm thinking gastropod. Very pretty.
Is that full zoom there?
Yes, it is.
Copy. Okay, come on. Let's see if we can get one a little closer to
Okay.
us straight down.
Coming full
Not sure if
wide.
we can or not.
That's full.
Copy. I'm just not sitting down. I think I'd squish one if I did. How about this one right here? Is that a different species there? Dead ahead? Come on, any
Okay.
video?
Zooming in.
Is that different, Brian? It is, isn't
Yeah,
it?
it
It's
is.
a different one.
Of course. Okay.
Same thing. Dead though.
Okay. Come partial.
Okay,
Video.
coming partial.
Hey, Scott.
Hi, Diva. Hi, Deb. It's Scott here.
That's
Hey,
partial.
feel free to give us your thoughts.
Yeah. See if I can get this one right over here.
Sure, my thoughts. So when I wrote down there Scleraxonian, like Paragorgid or Anthotheliid, what I meant was there's another genus called Parisis, a different kind of octocoral, which belongs
Okay,
to a
come on,
completely separate
That's
group
pretty good.
from the bamboo corals, and yet they do have nodes. So they do appear to be articulated, but the node is a different construction from bamboo corals. So this group called the Sclerexonia, they don't have a true axial skeleton. Early on, we were looking at one of those things we thought perhaps at first, well, I thought, I said maybe it looks like a Paragorgia, but when we got close, we could see there was some tissue eaten away, and you could clearly see the internal skeleton. So that's kind of like a proteinaceous, almost woody-like internal skeleton. Sclerexonians don't have that, and so if this is Parisis, it can have nodes, but it's got a completely different skeleton structure. And it's interesting, I don't usually work at these shallow depths, but Sonia Rowley was pointing out that the Parisis are more common as you get shallower. So that's certainly a possibility for this. Chris Kelly is noting that there is a dead skeleton nearby with a clear node, and I apologize, I didn't see that. But very interesting. This would certainly be something new for all the dives that we've had so far. And I'll keep looking, see if I can tell you anything more.
Great, thank you, Scott, and we really appreciate you calling in.
Do we want to take a closer look here, watch leads, or is that?
Yeah, I think one more close look would be really good. Thank you.
Sure.
I think what would be useful is if there was a colony that showed up that has like one branch that was completely dead, then I think see the possible nodes much more clearly.
Yeah, and if it's possible, I mean, it's difficult from this angle, but if it were possible to see it on more of a plane view instead of kind of above, that might be helpful, but I realize the corals are not necessarily cooperating with it.
Okay, be on the lookout for partly dead ones. See anything, Brian?
Not yet.
We do a partial here video.
Of course.
Hold there. Just look at all of these guys. See if we can find one with a dead
Copy that.
branch or something.
There's a branch on the ground on the top right.
Branch ground, top right. Okay. See any other ones?
Nope.
Okay.
There's something laying on the ground.
Yeah, up from lasers.
Hi, it's Sonia here. I'm pretty confident that these are Parisis. I do see this morphology and this coloration and very
there's another one right in front of
similar porous structure from about 140 to 150 meters on the mesophotic reef. So I'm very confident that's what we have here.
Great, thank you, Sonia. We really appreciate you getting on the line to let us know that, and that's helped to solve the mystery of these incredibly abundant corals.
Can we just go partial video?
Of course.
Is that one right? Hold there, right down there. Is that partially dead?
Maybe, yeah.
Okay.
Yeah, definitely possible. Could just be polyps retracted too, though.
Yeah. Come full in.
Full in.
Nope. Keep coming in.
Oh, I see.
Keep coming in.
Stand by. Full zoom.
Copy. Hold there.
I don't see any nodes.
Okay. And actually come wide. Let's see if we can get a better shot. Was that not what we were looking for, Brian?
No, I think that was.
Okay. Come full wide. Okay, come back in video.
That's partial.
Copy. And there's a crustacean over there. Okay, keep coming. Hold there. Is that full?
Not quite, I don't think. Oh, yes it is.
Okay.
I think that's good enough. I don't see any nodes.
Okay, so onshore scientists, what do we want to do? We've sort of successfully ID'd this animal, and so do we want to continue moving up slope, or we have about half an hour left in this dive?
Why don't we just continue up slope and see what else is on this really marvelous ridge?
Okay,
Great.
full wide video. Thank you.
Okay, pilots,
Full-wide
we would
pilot.
like to continue up slope, please.
Copy that.
I think 195 probably.
195, aye.
Bridge nav.
All right, spin around with me, and I'll jump up
Let's
there.
do 40 meters at 195 degrees at 0.2 knots, please.
While we're transiting, I'll just toss in that the name Paraisis, P-A-R ISIS, means next to, and so it's kind of the name is referring to the fact that it looks kind of like a bamboo coral, but it's not. So we'll say that it's next to the bamboo corals.
That's really interesting. Thanks, Scott.
Yeah, thanks for a little bit of etymology.
Yeah.
That's why they don't let me pilot.
Nav, how much time is left? I'm sorry, I've forgotten.
30 minutes.
Thank you.
We still got time for one more shark, I think.
Excellent. Well, I don't know about you, but I think this has been a really fantastic dive. I've really enjoyed the fact that we've just seen such an abundance of biology. We've seen several commercially important coral species. We've seen at least a few commercially important fish species. And then we saw that huge field of basket stars. Then we saw a huge field of corals. So it's been really, really interesting. And as Shirley Pomponi on our chat log points out, there have been these very distinct zonations. First, we saw a whole chunk of basket stars. Then we saw a whole section of corals. And now we're in yet another different type of coral, I believe. So it really has been. There were little zones of transition, I believe, but they really are quite dominant in each separate area. perhaps my favorite part of the dive from the biology standpoint was actually the weird sponges that we saw, those round things that were like geoidia.
That's the biology highlight for you.
I thought so. They were really cool. Well, okay, the basket stars were pretty cool too.
I'm just speechless. How could you not?
Okay, the shark was also pretty cool.
Exactly.
Okay, maybe it was the shark.
No offense, Shirley, but
maybe it was the shark.
The basket stars were just phenomenal.
The shark.
Shark.
It wasn't a 6 skill.
Yeah.
It was still very cool.
Still shark. Sharks.
I don't know if we installed the shark finder on Sirius yet.
We can't hear you at all, Nav. Say that again.
Is that why you didn't see it?
Yeah. Could probably find a couple more.
Yeah.
Yeah, that's true.
Also shark bait.
Nice. I'm gonna snap on this shiny looking guy here.
Stands out a bit.
So what is that?
That's a sponge, and I'm not quite sure what type. I know Shirley's still on the line.
I was going to say, if only we had 2 sponge experts. We do.
We do. Shirley and Chris, what kind of sponge is that? Looks kind of like a demo sponge to me. This is when they're going to chime in and be like, no, it's wrong.
It is a demo sponge.
Yes. Another win.
Good, take that, co-pilot.
And Shirley says, wow, it's a demo sponge.
Just give it one tilt down.
So why is that exciting?
I don't know.
And come on in.
Shirley, we're waiting. We're waiting.
Maybe it's
we
a
want to know
different
why it's exciting.
morphology than what she's ever seen
Oh,
before.
she thinks it's a lithistid, which
Lithistid.
I am. Yep, lithistid, which I am learning how to pronounce, which is the rock sponge that we were talking about another day. And the reason it actually came up the other day is because we were looking at a rock that had a lot of gas venting holes in it, and someone said it looked like a rock sponge.
Just to the right there, is that a coral that it's like on top of? What is that?
Not sure.
Do we want to hold this for a little bit, watch leads?
You're muted.
Yes, please. Hold on.
Okay.
So, shore-based scientists, is this something that's important to collect?
Bridge now.
Well, I'm biased. You know what my answer would be.
Please.
Is this a potentially a new occurrence or
Come up as I come under you.
something you didn't know was going to be here?
I think forget about the fact that it's a sponge for a second.
Right.
Let's look at it objectively.
Is it a novel
Just fill
specimen?
the whole thing here, video. Fill the frame. Yeah, whole thing.
I think Shirley might actually die of a heart attack before we're done collecting this. She seems very excited.
Scott's put if collecting urgency is based on punctuation. Okay, it sounds like we should
Bridge
collect
now.
this.
Copy.
So, pilots, we'd like to collect
Come on.
this sponge, please.
Let's do 10 meters, 10 meters at 015 degrees, 0.2 knots.
Pilot,
Thank
did
you.
you get that?
Sure did.
Okay, just making sure. All right, so this is going to be one of our, this one we've deemed interesting enough that we need to collect it. So this is actually going to be one that has 3 biology samples for this particular dive. And that makes a lot of sense because we've had so many interesting and novel occurrences today. So very exciting.
So because it's a potential lithistid, I think we can consider it a rock specimen, right?
No. Absolutely not.
I mean,
I was kidding. Well, I mean, we could consider it a rock specimen, but then we'd have to put it, you know, in a Ziploc bag without anything in it and then put it into a rock bag. I don't think it would do well. As opposed to the care that we take with our biological specimens.
All right.
Are we grabbing this puppy?
Yeah. We are. And I guess, well, let's go port. I want on the starboard's got a flight risk.
Yep, you want to go port enter.
Yeah. Spin me up there.
You're up.
Copy. And you can bring up Mini Zeus, please. And you might be able to extend it, actually. What else is in the port inner? Port outer? Something stationary?
Port outer is a black coral.
Copy. So yeah, you can, well, we can wait to do it after I get it. But
Okay, so we're going to collect this
arms live.
lithisted sponge, rock sponge, and we're going to find a nice safe place for it to come back to the ship with us.
Copy.
We collected some very nice imagery of it just a few minutes ago.
Yeah, you can come down to the left of the Mini Zeus. How squishy are we talking here, Deb? Or
Yeah, really? You're asking me?
I mean, I think
it's a rock sponge.
I think it could be quite squishy, so just be careful.
Shirley and Chris,
Please.
how squishy might this sponge be?
If it's a demo sponge.
Copy. So once I get it, if you just
Actually,
wanna
they said that the other demo sponge, the ribbon sponge, though, is actually quite firm.
Okay,
Yeah,
so
they're
she
not
says
squishy.
They're rock-like, so not squishy.
rock.
Of course, we've seen what you guys do to rocks, too, so
Hey,
don't squeeze too hard.
I'll take offense to that.
No, I didn't mean it that way. I'm going to stop talking now.
This is just like in the aquarium trade. We call this a live rock.
Oh, okay, so this should be
Let's go on the
pretty
port
firm
side.
when they go to collect it.
Port side.
Oh.
Okay. So let me squeeze a little harder than that. It is hard.
Yeah,
Thank you.
Shirley's now saying very firm. It's a rock underlined sponge.
Ooh, and she explained why. It has lots of interlocking spicules that make it rigid. So, Shirley, are these all silica spicules, or
Oh
are
my
some,
goodness.
or any of these calcareous, or
Got it.
what exactly is the makeup?
Yep. Quick. There you go. Hold there. Okay. Come down, Mini Zeus.
So, all of the spicules in this sponge are siliceous. I think you could have put that in the rock box.
Nice.
Nice job, pilots.
Yeah,
Thank you.
great job. Thank you, guys.
Yeah, thank you. Nice.
Package is secure.
Copy that. See if I can get this out of the light. Maybe in a little. I'll just leave it there. There you go.
I was kidding about putting it in the rock box. It actually went into a bio box. Don't worry, people.
Thank you, Copilot.
Yeah.
So that was our third great collection from our very skilled ROV engineer team. So far we've collected a black coral or Antipotherian.
All
We've
right.
collected a basket
Bridge
star
nav.
or Gorgonocephalid. And we've also collected a
40 meters,
lithistid
195,
sponge.
0.2, please.
So a lot to do later
Hydraulic extraordinaire over there.
once they come on deck.
Captain.
And we've only got about 10 minutes left on this dive, which we've seen pass through several different areas of different geology and different faunal assemblages to go with it. We're currently at a depth of about 310 meters at a site known as Zealandia Bank, where we were hoping we would see several commercial species of fish as well as coral. And we did just that. We accomplished both of our goals. We saw some deep water snappers, some roughies, and also some pink and red corals, or coralliums.
We're headed south.
Yep, 195.
Copy.
And science team, please remember we'll be doing our call pretty much directly after the ROV comes off bottom, probably about 3 to 4 minutes after that. We just need to switch out the phone lines. If you have been in the event log but not called into the telecon, it would be great if you could do that. So please start searching through your emails for the telecon line.
Which of these is not like the other?
Come on in, video. And hold there.
So what is that gold sponge?
So this looks like another one of the hard corals we saw earlier, the scleractinians.
Is it partially dead? What's on the right hand side of it?
No, so it's like, oh, yeah, maybe. Didn't see that until you pointed that out.
Sorry, come on, video.
Yeah, so it's likely a Enelopsimia, which is a type of dendrophilid.
Okay, come on back in.
I think the brown part of that is
Hold there.
a dead skeleton. Sorry.
Ooh, I was right.
High five, geologist friend.
Hey, Roland. Or whoever's over there, Tara. We're having problems with audio.
Okay, come on in, video.
Okay.
Nice shot.
That's gorgeous.
Okay, so my ID was wrong. It is a dendrophilid, but it is not Enelopsimia. It's actually Eguchipsimia. Yeah, I mean...
Yeah, nice. I was going to say this is a nice opportunity to compare the polyps of a hexacoral, the yellow coral that you're looking at, with an octocoral, the white one that's just next to it, and see how the tentacles are different, the thicknesses are different, the number of tentacles are different, and then on the octocoral there are little side branches on the tentacles that the scleractinian, the hexacoral, the yellow one, does not have.
Copy that.
That's really cool. They do look very, very different from one another.
Watch leads, 15 minutes of bottom time.
Well, I'm not so sure this is Aguchi Somnia. It's just is a lot more branching and looks almost like a more fuller colony. I can't really put it in words than the Aguchi Somnia I'm familiar with.
Sure.
90 degrees.
Copy that.
Shore-based scientists, I just want to let you know we have 15 minutes left. 15 minutes of bottom time.
Drifting a little to starboard.
I see that, yeah. Thank you. Not accounting for the wind.
Okay. Okay, thanks.
The underwater wind.
Just for the wind.
That's kind of cool when you look that way. It almost looks like dead ahead, the fans disappear.
Yeah.
And if you'll notice that all of the corals are oriented in the
Because
same
if you
direction.
go this way, they all.
Nope.
Yeah, that's cool. Oh, I shouldn't have done that.
Thank you, Bridge. Let's do another 30 meters, same direction, same speed. Thank you.
Probably.
The stuff that looks a little bit like rubble, is it actually rubble or is it something else? Looks like coral rubble from
Yeah,
up
it
here.
might be coral rubble.
Let's take a look.
Can we get a snap zoom, please?
I think it probably is, because when we were zoomed in on the
video.
yellow coral, there was definitely dead coral rubble around there too.
All there?
Yep, lots of dead coral.
Graveyard. Okay, we can come live video. Just to see the whole thing here.
So one interesting thing here, if these white colonies are parasis, as we think they are, or paresis,
Can we snap right
then
down
I would think that their skeletons would fall apart not too long after they're dead. And so these other ones would presumably be different kinds of corals, maybe those yellow stony corals that we were just looking at.
Okay, you can come wide. Don't know what that is.
So Scott, why would they break up so quickly after they die?
That's because the skeleton is not made of calcium carbonate the way the stony coral was. The yellow one, again, is a very solid calcium carbonate skeleton. There's not a mystery why they're called stony corals. These ones are more like cemented sand grains, and so they don't hold together quite as well. And there's a range within the Sclerexonia, the group to which these white corals belong, so some of them are stronger than others. In fact, I should be careful because corallium, the precious coral, is also in that group. And, of course, they have really well-cemented sclerites. They're still cemented. There's no actual skeleton in the sense that it looks like that sort of wooden branchy coral that we were looking at before, the typical sea pan. But it's so hard that it can be polished and used as jewelry. So I guess some of them are hard and would last a long time. So maybe I'm actually wrong about the Parisis here.
Oh, it was at least a good theory.
Hi, Scott. Yeah, I'm in two ballparks here. I think there's something you mentioned earlier. I think we do probably have two different things with these white colonies. Even though some of the Parisis that I do bring up from the deeper mesophotic depths, they really are quite tough. I'd be surprised if their skeletons sustain, what we're seeing here in the dead material.
So, Sonia, do you think then that the skeletons that you're referring to from the shallower waters, they would remain for quite some time after the tissue dies?
I'm not sure they would remain as long as the sclerites. Yeah, that's for sure.
Yeah, I think I'd agree with you that either way.
Also, I'm now in the EEC here, and noticing the polyp structures of those ones, particularly by the end of Samlia, I can never pronounce it. They were really quite different.
Yeah, absolutely. Yeah, and if you need to go further, go for it.
Sorry, just to carry on, I wasn't working the thingy out right. Quite different to what I would expect for a Parisis. So I don't know what your experience is there, but I'd certainly be interested. There are different alternations that I would be seeing.
The one that was next to the sponge that we collected, I noticed at the base or just to its left, I couldn't tell if it was one colony or two colonies next to each other, had really large swollen nodes, just like you would see on a Melotheid. But otherwise, the colony didn't really strike me as a Melothea, but that just made me wonder if there were actually two different colonies that we're seeing, or two different species, that we're seeing down here.
Yes, Scott, I agree, actually. It's quite torturous.
So are these some fish? Yeah, coming up. If we could just get a quick zoom on the fish around.
Go ahead, video.
So, kind of one other question for the geologist. Did we decide earlier that this is a drowned reef, so carbonate? And if so, are some of the pits and things that we're seeing some of the old coral reef that was here? And is there any possibility that these are very old skeletons?
So this is another one of the deep water snappers, another Etelis carbunculus.
Let's do another 20
So this is another great
same
observation, as well as there's a shrimp underneath the ledge.
There you go.
Scott, you asked if this had evidence for being a drowned reef. As far as I know, I don't really think we saw any particular evidence that would have given us a clear indication that it was so.
Great, thanks, Deb.
We can zoom back and get the tail on this just to be sure which species it is. We're looking for a dark spot or none, or dark blotch or none on the upper lobe of the caudal fin to tell the two species apart.
Okay. Going in. That's cool.
That's good, and it doesn't look like it has a dark upper lobe, tip to the upper lobe of the caudal fin, so Etelis carbunculus it is.
Just give me a tilt down, co-pilot.
Very cool.
And Chris has identified the shrimp underneath the ledge as Placionika. And Bruce has said that perhaps these appear to be quite small, so perhaps they're juveniles of Etelus carbunculus.
Just a tiny
Great,
bit
thank you,
down.
pilots.
This
We're gonna...
This species of deepwater snapper tends to hang out very close to the substrate and also is usually associated with shelter, like under this ledge. That's why he's hanging out here. One possibility is that it is a smaller species of snapper and it's subject to predation from things like large jacks and other fish like that. The larger species of Etelus coruscans actually schools and spends most of the time well above the substrate. But the one time it is close to the substrate is when they're juveniles that are about the same size as this guy. And then at some point they get large enough where they feel comfortable getting up off the bottom and away from shelter where they can resume an adult lifestyle.
You say a couple minutes, a couple more zoom, one more zoom?
Yeah, one more.
Yeah. Copy.
Watch leads, navs.
And Copilot, you can start to come up a little bit.
We've got time for one more good zoom on something. If you see anything in the field of view, and then we got to get going.
All right, Shore Base Science Team, we've got time for one more zoom. Find something fun. Anything in particular you'd like to see just one more time?
There's something sort of striped dead ahead in the center.
Striped dead ahead in the center.
Just a series of sponges or something next to each other, maybe? I don't know.
I can turn to you at the end, too.
Yeah, go ahead.
Equipment starboard.
I don't think I see what's
I think it was just a row of sponges.
Oh. Anyone? Anyone?
I think... Has anyone suggested anything?
We tried something, but we couldn't find it.
I think we should just zoom, and because there's always been little tiny animals amongst the coral rubble, like little
What's the yellow
pagurids.
thing in the center
No,
left?
that's one that we don't want to look at.
Oh, we
that.
already looked at that?
No. That's another one of those hard corals that meh.
Okay.
So I think if we just zoom on a patch of...
Okay, zoom.
Just zoom. See if we get lucky.
I think that's a good idea, Diva.
I agree, Diva, because there's going to be lots of stuff encrusting on that. On the,
So here, there's, I mean, I guess these look like sponges or tunicates, right, Shirley?
Like what's all those little blue, Well, or little patches of sponge as well. I bet you there's like lots of little encrusting sponges. I'm not sure. Ooh, can we go in closer on this?
A couple little bits of dead coral for everybody.
There may have been another Sidarid urchin spine to the right, off-screen right.
I mean, look at all this biology, everybody.
In a little tiny space. Oh, look, there's a tiny little gastropod, I think. Or no, it's a hermit
Look,
crab.
there's a pagurid. See? He's right down at the bottom of the screen, walking along.
I've got a little more zoom I can take.
Is that a hermit crab? We had several requests for close-up to hermit crabs earlier. This one off the bottom screen center.
That's pretty small.
It's so hard.
Somehow I feel like this means that we're done.
I think that
was a great zoom. Thank you very much, pilots, and thank you for navigating an awesome dive, Brian, and for skillfully collecting 3 great specimens, pilots.
All right, so Shore Base Science Team, we will talk to you in just a few minutes. We just need to probably switch the audio line. So
come up where we are now.
We'll be back to chat with you in just a few.
I don't know, 100 meters or something, just way off. Push out a little. As I do, of course, there's a fish out there.
I'll hold real quick.
You can just keep coming.
Okay.
Yeah. Donezo.
Copy that.
And it looks like there's some pretty strong current layers out there, so we're going to get thrown around a bit in the short little bit of time we have to get to the surface.
You want to go snap your video?
Sure.
Boom. There's fish.
Oh, just as I'm signing off, this is another one of the commercially valuable species. In Hawaii, they call this a gindai. It's the oblique-banded snapper. Christopomoides zonatus. I'll see if I can get to a keyboard to type that in, or maybe Chris can do it. So this is another one of the bottom fish species that we were looking for.
In the background, it looks like.
You can hold
What did it say? Oblique-banded what?
Oblique-banded snapper, and Chris has the scientific name in the chat room.
Yeah, Chris got it. Okay.
You can keep coming.
There's actually some bigger fish even beyond it. There's more of them.
Oh, no. Keep out ahead end for now.
Okay. Yeah, because I want to turn left.
So we just barely got into bottom fish territory on this dive.
Okay. All right.
Hold there.
Yeah, hold there. And you need to come left, you said? Oh, no, I think I just need to come around, right? You need to come left. You go ahead and align yourself with the ship, actually.
See you later.
And
catch you on the flip side.